Every physical trait you possess, your eye color, your height, your hair texture, traces back to instructions buried deep inside your cells. If we zoom all the way down into your chromosomes, we find the exact molecules storing those instructions, the DNA double helix. Look at this chain forming on screen. DNA is a polymer, meaning it is a massive molecule built by repeatedly linking together smaller identical units. Think of it like a freight train, where the long chain is made of individual boxcars. In DNA, each of these individual boxcars is a monomer called a nucleotide. To master this topic for the WBCHSE board exams, you have to analyze this molecule through two distinct lenses, as shown here. On the left, we have the chemical structure, the raw microscopic ingredients that make up each nucleotide. On the right, we have the morphological structure, how those ingredients link and fold into a physical three-dimensional shape. You cannot memorize the shape without first knowing the parts. We have to identify the exact chemical ingredients before we can understand how they build that famous twisted ladder. Let's isolate a single nucleotide and split it into its three chemical components. Every nucleotide starts with a pentose sugar called deoxyribose in the center. Attached to one side is a phosphate group, and attached to the other is a nitrogen-containing molecule called a nitrogenous base. The sugar and phosphate are identical in every nucleotide, but that nitrogenous base comes in 4 variations, divided into 2 families. Look at this first branch on screen, the purines. This family includes adenine and guanine. A quick way to memorize this for the exam is the phrase pure as gold. P for purines, A for adenine, G for guanine. The second family branch contains the pyrimidines. In DNA, these are cytosine and thymine. We now have our complete chemical alphabet, A, G, C, and T. The sugar and phosphate link together to form a rigid, uniform scaffolding. They never change, but those 4 nitrogenous bases act as the variable sequence, the actual readable code of life. Now we assemble the morphological structure. Notice how these nucleotides link vertically. The phosphate of one binds to the sugar of the next via strong covalent phosphodiester bonds. This creates a single strand. To make the complete molecule, a second strand aligns next to it, but it runs in the exact opposite direction. This opposing alignment, one side pointing 5' to 3' and the other 3' to 5', is called being anti-parallel. The 2 strands lock together across the middle through complementary base pairing. Adenine only ever pairs with thymine, and they are held together by exactly 2 hydrogen bonds. Cytosine only pairs with guanine, locking together with 3 hydrogen bonds. Once the entire ladder is paired, the natural chemical angles force the structure to twist into a right-handed spiral, the iconic double helix. This physical twisting pulls the variable, readable code into the center of the molecule, wrapping it in the tough sugar-phosphate backbone. The shape itself shields the fragile genetic instructions from damage. DNA is locked inside the nucleus, so to build proteins out in the cytoplasm, the cell needs a mobile photocopy. That brings us to RNA, ribonucleic acid. To make this copy, the cell applies 3 structural rules. First, as we see here, it splits the ladder and builds a temporary single-stranded molecule instead of a double helix. Next, it makes 2 strict chemical swaps. It replaces the deoxyribose sugar in the backbone with ribose. Then it entirely discards the base thymine, replacing it with a new pyrimidine base called uracil. Let's test that logic. If the original DNA sequence is ATGC, What is the corresponding RNA sequence? Since adenine normally pairs with thymine, but RNA uses uracil instead, the sequence becomes UACG. The single-stranded shape and the presence of the ribose sugar make RNA less stable than DNA. It degrades quickly, which makes it an excellent short-term messenger that won't clutter the cell once its transport job is done. For your exams, you need to synthesize all these facts into a formal comparison. We will build a standard 5-point table contrasting DNA and RNA right here. Row 1 is morphological. DNA is double-stranded, while RNA is single-stranded. Row 2 is chemical. DNA uses deoxyribose sugar, and RNA uses ribose. For row 3, DNA relies on adenine, thymine, guanine, and cytosine. RNA swaps out thymine for uracil. Row 4 details their location. DNA is trapped inside the nucleus, whereas RNA moves freely between the nucleus and the cytoplasm. Finally, row 5 outlines their function. DNA acts as the permanent storage of genetic information, while RNA executes the instructions to synthesize proteins. You'll want to pause the video and screenshot this completed table for your notes. Mastering how the chemical components build the morphological shape allows you to understand the exact differences between these two molecules. That conceptual logic ensures you don't just memorize the table, but clearly understand it to secure full marks.